Hi, my name is Jean-François Millot. I am CEO and co-founder of Tatum Bioscience, and I'm going to show you how we are cracking cancer resistances using a new modality. Immuno-oncology revolutionized the way we treat cancers, but is far from having reached its full potential. Very few patients respond to treatments because most of the time immunotherapies fail at igniting the anti-tumor immune response. In parallel, many of them develop adverse events because immunotherapies are administered systemically, triggering non-specific immune responses which are toxic. At Tatum, we propose an alternative approach. We have developed a new modality based on engineered phages that multitask immunotherapies. This drug is directly injected in the tumor and triggers an immune response specific to the cancer cells, which greatly decreases toxicity. This multi-pronged immunotherapy activates the innate immune response, the T-cell response, and the B-cell response, all of which circulate the body of the patient to not only eliminate the treated tumor, but also the metastases that share the same antigens. At Tatum, we are leveraging this technology to build a platform to crack cancer resistances. Our therapeutic phages are produced using a bacterium that we can program with the therapeutic DNA coding for the therapeutic agents to display on the phage. This gives us access to a wide array of phages with different combinations of therapeutic agents. We can then test those phages on different cancer types and find for each of them the right combination that overrides resistances and triggers a potent anti-tumor immune response. To demonstrate why our technology is superior, we've performed a benchmarking experiment in solid lymphoma model which is resistant to PD-1 and PD-L1 checkpoint inhibitors. As expected, the efficacy of Roche anti-PD-L1 is quite limited on this lymphoma. However, if we treat this cancer with CNAB-03, which is a candidate combining 4 therapeutic agents, an undisclosed molecule, an antigen, a TLR9 agonist, and Roche atezolizumab, then we override the resistance and clear 100% of the tumors. This shows that in immuno-oncology, you need synergies between multiple therapeutic agents to break cancer resistances and create the right environment to trigger potent anti-tumor immune responses. Regarding the competition, we've divided it into 2 groups. The first one, we group single-activity immunotherapies. And as our data demonstrate, a single activity is not enough to create the right context to break cancer resistance. Instead, we believe the future in the field is with multi-pronged immunotherapies. In this space, we are competing against RNA-DNA technologies and oncolytic viruses. How do we differentiate? Both technologies rely on the cancer cell they aim at eliminating to produce their therapeutic effect, which we think is not the optimal strategy. Tatum is thus best in class because we can multitask immunotherapies, we are not dependent on cancer cells, and our drug activity is instantaneous as soon as we inject it. Tatum is supported by an amazing team, starting with my 2 co-founders, who are renowned experts in synthetic biology. We are also very fortunate to have incredible advisors. For instance, Dr. Howard Kaufman, who is the former president of the Society for Immunotherapy of Cancer, and Professor Jamie Scott, who pioneered phage display in the early 90s, from which our technology is derived. To bring Tatum to its next development phase, we have a 6.5 seed round open to nominate our lead candidate, with 1.5 million in the process of being secured. We are also looking for a pharma partner to co-develop candidates. So feel free to get in touch with us if you are interested. Thank you very much.